Awesome. Thanks, Jim. Thanks for inviting us to this conference, and thanks to Daniel Powell, with whom I'm collaborating on this project. He's given me the honor to debut this for you. This is ultimately the other way, Jim. This is ultimately a culmination of something I've been working on for about the past 2 decades. So as Jim mentioned, I'm with Spark Biomedical. Spark is working on and has on market a auricular stimulation device. Jim, if you wouldn't mind giving me the next slide. An auricular stimulation device for that stimulates cranial nerves in and around the ear that has received FDA clearance to mitigate the symptoms of opioid withdrawal in adults. The system is called the Sparrow. What's pictured here is actually the Sparrow Ascent, which is a second generation system that is not yet FDA cleared, but we're getting real close. Never been close. About 9 months ago, Spark formed a subsidiary company called FiveLiters. Spark and FiveLiters have licensed from Northwell Health here in the New York City metropolitan area, partnered with them in FiveLiters to commercialize something that's called the neural tourniquet. And Dan and I and others on the team are looking very carefully at the multiple points of market entry for this potential technology. I'll walk through that a little bit, but first I want to give you a little bit of glimpse of what the neural tourniquet is. Jim, next slide, if I may. Thank you. Oh. Jim, next slide, please. There we go. That's okay. That's all right. So I'm going to take this story back to the beginning. In the late 90s, actually, let me go back a little further. In the mid 90s, there were other groups of researchers who were very interested in how the brain senses infection and mounts fever responses. And what these groups had figured out back then was that products of the immune system and products of bacterial pathogens trigger signals up to the brain via the vagus nerve. And the brain responds by mounting fever responses and behavioral changes like lethargy and food avoidance and things like that. A few years later, we were working quite accidentally in a different space, and we discovered that not only does the brain mount these central responses to an infection, it also mounts efferent signals down the vagus nerve that ultimately culminate in the spleen and regulate inflammation. And it does that in part by educating immune cells in the circulation that rather than mounting a robust inflammatory response, it's time to mount a healing response. So these neutrophils, monocytes, are educated by this vagus nerve signal. Dr. Lerman kind of referenced some of this in his talk. And so round about 2000, we said, well, okay, if monocytes and neutrophils can be educated by the vagus nerve, what other cells might be educated? And we thought, well, maybe platelets is one of those things. So we partnered with DARPA. And with a $5 million grant from DARPA, we went on to discover this thing called the neural tourniquet. Jim, next slide. And what we showed initially in normal mice and normal pigs is that application of stimulation to the vagus nerve, 5 minutes or so, is sufficient to reduce blood loss to injury by about half. And we kind of poked through this, trying to understand a little bit better how this might work. And it turns out now that application of vagus nerve stimulation sends a signal to the spleen and educates the platelets, much like it educates neutrophils and monocytes. And that happens through release of acetylcholine in the spleen. The circulating cells pick up that acetylcholine, and it essentially changes the phenotype of those cells. We can measure the effects of vagus nerve stimulation on bleeding in a number of ways beyond just blood loss. We know that the intracellular levels of calcium go up within those platelets. We know that in the wound, if you look at blood coming out of the wound site, you have greater production following VNS. You get greater production of thrombin in that shed blood. Thrombin is a very important marker of coagulation activity. So we know coagulation activity is going up specifically in the shed blood. This isn't happening systemically. It's happening specifically at the wound site. Those platelets, when they see a thrombin challenge, which is being generated in the wound normally, they have a much more robust response. They release alpha granules, and you could find P-selectin on their surface. And you can measure systemically in the blood. You can use a technology called thromboelastography, and you can see more rapid activation of coagulation ex vivo in that blood. We know that the acetylcholine receptor the platelets express is required for this pathway. We know that neural innervation of the spleen is essential in this pathway. And we know that there's this very specific T cell in the spleen that's required for this pathway as well. And Jim, next slide, if you may. The other thing that we've now since figured out is that, and this is now, it's papers, we just had a paper accepted to Nature Communications that shows that this pathway is actually independent of factor VIII. Factor VIII, coagulation factor VIII, is the genetic defect present in hemophilia A and is one of the central clotting enzymes in the coagulation cascade. And what you see in the data plot on the left is blood loss in either wild type animals. This is a mouse model where we do partial tail amputation. Wild type animals on the left side lose a certain amount of blood when you snip off part of their tail. If animals are knocked out for factor VIII, that essential coagulation enzyme, they lose significantly more blood from that wound site. But application of vagus nerve stimulation, in this case, 5 minutes of stimulation, I think it was 15 minutes before tail snip amputation, reduces their phenotype back to wild type. So these these factor VIII knockout mice, they are back to losing normal amounts of blood rather than the exaggerated amount of bleeding that you would see in hemophiliac. In the beakers on the right, what you see, these are hemostatically normal rats that either received sham stimulation or vagus nerve stimulation prior to a liver puncture model. So this is sort of a non-compressible hemorrhage model. And we see that vagus nerve stimulation significantly reduces the amount of blood loss from those sorts of wounds. Jim. So a lot of the work that we had done was in hemostatically normal mice, rats, and pigs, which opens the opportunities for things like trauma, surgery, kind of routine use of vagus nerve stimulation for bleeding control in those situations. But this more recent work that we've done shows that it works in the context of hemophilia, and it really opens the door for a lot of the bleeding disorders that are out there now. So Dan and I and others at Spark are working very closely trying to figure out, of these opportunities, where do we go first? Jim? And one of the things that we are most interested in are the bleeding disorders. These are really complicated disease states. The treatments that are available are adequate, though extremely expensive. It can cost $300,000 or more for an annual course of treatment for these diseases, and that's just their primary course of treatment. These patients also have a lot of comorbidities, so their healthcare costs far exceed that. So there's a lot of potential opportunity there. We are envisioning vagus nerve stimulation through a version of the Spark device as a adjunct to standard care, so that this is something that they could potentially use to reduce their bleeding events. And next slide, Jim, please. We're also looking at von Willebrand's disease. So von Willebrand's disease, hemophilia, especially hemophilia A, and even more so hemophilia B, are relatively rare diseases. Von Willebrand's disease is much more common. There are up to 3 million people or so in the United States that have von Willebrand's disease. Most of them are of rather minor or moderate clinical severity. And there are therapies available for these subjects or for these patients, though they're not necessarily the greatest of therapies. They do have their issues, and they are incompletely covered in terms of their ability to control bleeding. So much like within hemophilia and von Willebrand's disease, we also see this as sort of an adjunct therapy that would go alongside existing therapies. And last slide, Jim. So currently where we are today, we began this project 9 months ago. We have 3 clinical studies underway right now. There's a healthy human subject study underway, or just starting up with Northwell Health. There's a lumbar spine surgery study that we've started. Those are patients who are, from a hemostasis or coagulation system perspective, they are healthy, normals. and we're looking at blood loss in that context. We're also looking at patients with infected dialysis ports who are undergoing surgery for those ports. These tend to be renal dysfunction patients who are uremic and therefore have platelet dysfunction. So that's a very interesting study. And just a few moments ago, we submitted a grant to the NIH looking at auricular nerve stimulation in the context of von Willebrand's disease. So hopefully, Nick, we get some success with that. And we're looking at pursuing breakthrough designation for this technology. And with that, I'll take any questions.
All right. Do we have any questions? Yes, Victor.
You mentioned about the learning, and you mentioned that you give VNS about 15 minutes before the trauma. Have you pushed the learning? I mean, how far does the learning go? How far in advance can you provide VNS?
Great question. So yeah, we have done that. It looks like the effect, so a short stimulation period, 5 minutes or so, seems to have a hemostasis effect that can last for about 48 hours. It seems to be gone at 72 hours. From a therapeutic application perspective, what we're thinking is something along the lines of, daily application, daily stimulation, 5 minutes, 30 minutes, once a day. I'm talking like in the bleeding disorder application. Obviously, somebody's going to undergo surgery. One can easily think about during pre-surgical testing or at some phase just prior to going into the operating room, apply stimulation for a period of time for the surgical use. But even in trauma, we have some evidence that suggests that in the context of active bleeding, the effect seems to take hold within we haven't been able to do the experiment shorter than about 5 minutes, but within about 5 minutes, you get a pretty full effect of this application. So there are a lot of platelets going through the spleen with every beat of the heart, and they're picking up this signal and very rapidly disseminating to sites of injury. So it even seems like in that trauma setting, you could come in and stimulate a patient who's actively bleeding and get a therapeutic effect.
Great. A couple of questions. Ron.
Thank you. There's so much beautiful science here. I know you said you're adapting the existing Spark device, but do you need to produce a very robust, vagus response? Does that mean you need to update the device? Do you think you'll need to update the device to get more fibers or something like that?
No, I wouldn't go that far. I don't think that functionally the device that we'll use, for example, in the context of bleeding disorders, would really functionally be that different than the existing Spark technology. I think we're looking at some different embodiments, but it's really not about stimulation paradigms or anything like that. It might be duration for opioid use. The duty cycle of the device is much longer, right? Patients wear that for a longer period of time. But in terms of stimulation parameters, I don't think they're really going to be any different.
Just in terms of placement of the device, it's placed on the cervical VNS. But you've worked on this for years, I presume. So have you looked at different places along that nerve pathway and places that might be to implant on that innovate different things? Or is it critical that it's up there and is actually affecting not just the spleen, but also the heart? I'm not sure that we've done that in the context of bleeding. We've done some of that work in the context of inflammation, but this device will be an auricular stimulation device. It's a wearable. Gotcha.
So it's not being implanted anywhere.
Wonderful. That's even nicer. So we don't all have to walk around with implants. We can just pop them on in trauma. Sorry, I missed that bit.
No, that's okay. And that's actually one of the reasons I'm so excited to be working with Daniel and the rest of the team at Spark is that, when we began this work 20 years ago, we kind of stumbled across this. It was frankly quite accidental. But we were using electrical vagus nerve stimulation as a tool to probe the system, not necessarily thinking that was the therapy end game, right? And especially we were really a sepsis lab and a bleeding lab, and an implant is, aside from bleeding disorders, an implant is not really a solution for sepsis or for traumatic bleeding, right? That's just not, or pre-surgical. So it wasn't until Spark came along with a technology that we can now robustly stimulate the cranial nerves in and around the ear. All of a sudden, these applications are available. So
the animal studies, have any of those they have been done now as wearables? Because I know all your past history is on the implantables on the animals.
All of the preclinical work that we have to date is with invasive VNS, very acute experiments, and with hook electrodes around the vagus nerve in the neck. So we're making two leaps, right? We're making species leaps and we're making a leap from site of location. So.
And if I could add to that, that's the purpose of our healthy human subject study.
Exactly.
Tease out all that without complicating it with this.
Thank you.
Okay.